Welcome everyone to the AIHM Friday Wellness Webinar. We're just thrilled to have Dr. Heather Zwickey with us today, and I'm just going to take a few minutes as everyone starts to come into the Zoom meeting. I'll welcome everyone on Facebook as well. Can you advance the slide? Thanks so much, Jess. So we are the Academy of Integrative Health and Medicine, a global interprofessional integrative health association working to transform healthcare, body, mind, spirit, community, planet. So if you're joining as one of our members, welcome, or one of our fellows. We are really bringing together the integrative community and recently merged with the Academic Collaborative of Integrative Health, and we're really excited to be working alongside of ACIH and that community as well. So I just want to welcome everyone as you're coming in. If you have not yet subscribed to our channel, please go to youtube.com/AIHM Global, and you'll be able to get the videos of these if you're not able to attend our webinars that happen every week at noon on Fridays, noon Pacific time. So again, welcome everyone. We have a lot of folks coming today, so we're going to take a couple minutes to let people come in. And I'm going to take a moment to welcome our special guest and a dear friend and colleague, Dr. Heather Zwickey. Heather has her Ph.D. in Immunology and Microbiology with a focus on infectious disease and vaccine development. She's a respected AIHM Fellowship faculty. She teaches nutrition and research and holds private webinars for AIHM Fellows. And she was recruited to the National University of Natural Medicine and UNM, where she launched the Helpcott Research Institute and established the School of Graduate Studies, developing programs in integrative medicine research, nutrition, and global health. And just has been such a leader in our community around research, and really a true collaborator and mentor to students. I do just want to give a little shout out to Heather, how you've just inspired so many students to get involved in research, to learn research, and to go out there and really be advocates for science-based, evidence-based naturopathic medicine and integrative health, and really have helped create an entire cadre of new researchers in this field.
Thanks.
So welcome. I'll just take a couple more minutes while folks are coming in. For those of you who are attending today through the Zoom, if you are interested in our fellowship, we'll be sending a $500 discount if you decide to join the fellowship. So we have a cohort that's going to be starting in April. So if you're interested, please let us know, and we'll email you after the event. And just to, before we have Heather start, just to welcome, we welcome questions. We have lots of questions that have already come in from folks prior to the event. So we're looking to listen to Heather's presentation and take questions at the end. Please use the Q&A feature for questions to the presenter. Otherwise, they kind of get lost in the chat. So you can use the chat feature for comments or reflections. If you can change who you're sending the chat to. So if you want to choose all panelists or send just to a specific person like Stevie Nicks, then you can choose that person and send them a chat. But
Stevie Nicks is here. I'm excited.
But if you would like to ask a question, please put it in the Q&A. We'll try and get to as many questions as possible, but I have lists of questions that have also come in prior to the event. So again, welcome Heather, and I'll let you get started.
Hi, everyone. So glad you could join us today. So we're going to talk today about COVID vaccines, and there's a lot of interesting information out there. So I'm just going to jump right in and get going here. These are our topics. We're going to talk about the different types of vaccines that are out there and then go through the specific vaccines, the data for each of those vaccines, and the implications. Then we'll talk about the safety considerations. And I know that's where a lot of your questions are, so we'll focus a lot of our time there. We'll talk about the public health aspect of vaccines, and then we'll go ahead and start going through all the questions that Tabby has accumulated. So let's start with an overview of vaccines. There are 5 major categories of vaccines that we currently have on our market in the United States. The killed or inactivated vaccine is exactly what it sounds like. It's a vaccine that is either killed by heat or formaldehyde. And currently, none of the COVID vaccines are heat inactivated or formaldehyde inactivated, because when we tried to make a coronavirus vaccine, specifically SARS vaccine, that was an inactivated vaccine, we saw something called antibody-dependent enhancement, meaning it actually increased uptake of virus instead of decreasing uptake of virus. So we haven't seen any of these hit the market yet, and I don't know if we will. The second type of vaccine is called an attenuated vaccine. And this is a type of vaccine where we actually use the virus itself, but we make it so that it is replication deficient and it is not pathogenic. And some of the attenuated vaccines will actually come out in the market. There's one being produced in India right now. The vaccines that are closest to market or on the market include a recombinant subunit vaccine. This is where we make a protein in the lab that is a part of the virus, but it is not the entire virus. It's a single protein. The vectored vaccine is where we take DNA or RNA from a virus or bacteria, and we insert it into a vector, another virus that is not as infectious, not as dangerous. And we'll talk about that. And then the RNA and DNA vaccines. And these are, of course, the vaccines that we currently have. So Moderna and Pfizer are the RNA vaccines, and those are on the market already. We've heard Johnson & Johnson has now submitted to the FDA as of this morning. AstraZeneca is also the Oxford vaccine. So this is the UK version of what Johnson & Johnson has done. We'll talk about that. And we'll talk about the Novavax and Sanofi vaccines as the recombinant and subunit vaccines. Let's remember that when we vaccinate, there is an ideal response to have. Our immune system is in balance between what we call a Th1 response and a Th2 response. A Th1 response is the type of response that you want to have to a vaccine. It activates macrophages, cytotoxic T cells, and natural killer cells, and that allows us to eliminate viruses and intracellular bacteria and tumors. And so what we want is a Th1 response. Now, there is such thing as a Th2 response, and this is the type of response that we want if we have parasites, things like intestinal worms. However, a Th2 response also is our allergic response. This gives us lots and lots of antibodies, but it doesn't give us the T cells. So if we're thinking about our response to a vaccine for a virus, we want to have a Th1 response. We want a strong Th1 response over a Th2 response, which means we may or may not generate antibodies with the vaccine, because what we're more concerned about is T cells. T cells are going to be what fight the virus. Antibodies are helpful when the virus is extracellular, when it's outside the cell, but once the virus gets inside the cell, you have to have a T cell to kill it. Current vaccines do generate an antibody response, but as we will see, they are also generating a Th1 response. So this was a paper that came out quite a while ago now, although, you know, historically, we wouldn't have said 6 months ago was a while ago, but these days we do. And this gives us a great model of the SARS virus and what we're making the vaccine to, which is the spike protein. So if you look here, this is a model of the virus. We've blown up the spike protein. And what you can see with the spike protein is that there is a receptor binding domain. Some people some scientists are making vaccine to the entire spike protein. Others are making a response just to the receptor binding domain, and others are making a response to the entire virus. So we've got different strategies. I already gave you the list of types of vaccines that people are working on. And then we've got the process to get to market. And I think we are all quite aware at this point that you can make the vaccines and do the clinical trials and get them through the FDA, but if you can't figure out how to get them to market, you're not going to have appropriate immunity. So one of our steps is slowing us down. It's the rate limiting step right now, which is the getting things to market. So as of March of last year, so about 11 months ago, we knew that these were the vaccines that were in development. The Johnson & Johnson vaccine, which is an adenovirus vectored vaccine. Here's that live attenuated vaccine I told you was being made in India. There's several recombinant subunit vaccines being made in Australia, the Novavax vaccine. There's a trimer. So what these folks are doing, and this one will hit the market here sometime, probably in May or June. They actually use 3 different spike proteins and hook them together. So this is a trimer vaccine. Here's another vectored vaccine. And we've got an oral recombinant vaccine that is being developed. So there will be an oral vaccine that comes to market at some point. And then we've got the DNA vaccines and the mRNA vaccines. Now, fast forward to now, February, and what do we have? Well, the vaccines that are on the market are here. And you can see that we've got the Pfizer vaccine, the Moderna vaccine. There are vaccines in other countries that are not on the market in the United States, like the Sinovac vaccine in China. This is the AstraZeneca vaccine, which is currently on the market in the UK, but is coming to the US. And Sputnik actually just released all of their data this last week. And they are also likely going to try to sell that vaccine in the United States. There are also 93 vaccines in the pipeline. That's a lot of vaccines. So Johnson & Johnson, Novavax, Casino, there's vaccines out of Australia and all over the planet that are in various phases of research. When things hit phase 3 is when we start to say, oh, they might make it. So be aware that there are lots of different vaccines and different styles of vaccines that are coming. And different vaccines stimulate different types of immune responses. So we're going to examine the types of immune responses that we're getting from the various vaccines. Here's where we are right now with efficacy. We know that the Pfizer vaccine and the Moderna vaccine are giving us roughly 95% efficacy. Now, what is efficacy? Efficacy is the ideal. In a controlled trial, when you're comparing to a placebo or comparing people who have been vaccinated to those who haven't been vaccinated, in an ideal setting, can we look at the attack rate in people who have been vaccinated versus people who haven't been vaccinated? This is different than effectiveness. Effectiveness is when we actually look at things in the real world setting. And so even though we have 95% of efficacy, we may not have 95% effectiveness. And I know there's a lot of questions that happen between efficacy and effectiveness. And so I just wanted to explain that scientifically they're different. Johnson & Johnson's efficacy is about 76%. If you throw in some of the data that's come out of the UK with Johnson & Johnson, it actually lowers this number a little bit. And then the Novavax vaccine is currently showing up at 89% for efficacy in an ideal setting. Let's look at this Pfizer vaccine first. And remember, our Pfizer vaccine is an mRNA vaccine with liposomal nanoparticles. So let's start with talking about how mRNA vaccines work, because I think there's some confusion out there on how these vaccines work. We take a piece of mRNA Usually we isolate that mRNA from a virus, and then we use what are called primers, where we manufacture these primers in the lab just using the basic components, and we grow using PCR lots and lots of mRNA. So we do that in the lab. It's not being isolated from fetuses. It's not being isolated from dead people. We actually make the mRNA in the lab. Then we surround this mRNA with fat molecules. And it's really easy to get the fat molecules to surround the mRNA. All you actually have to do is put them together and then add some ultrasound. So vibration, and the fat forms these little micelles around the mRNA. Now, the liposome nanoparticles, the combination of nanoparticles is proprietary, but the vaccine companies have released all of the information of absolutely everything that's in the vaccine, and I'll show you that in a minute. So once we've got this mRNA vaccine, we inject it into the deltoid muscle, and it's taken up by macrophages. It's not taken up by muscle cells. It doesn't make it into sperm. It doesn't make it into eggs. It's taken up by macrophages in the deltoid muscle. These are the phagocytic cells. Muscle is not phagocytic, so it doesn't usually take up the vaccine. And liposomes just don't automatically go floating around in your body. They have to be phagocytosed. So we phagocytose the liposome. It gets the mRNA into the macrophage, and the lipid is actually what's acting as the adjuvant. The lipid binds to what are called pattern recognition receptors, specifically a toll-like receptor 3 or a toll-like receptor 7 in the endosome. We release the mRNA into the cytoplasm. Here's where there's another misnomer that happens. People are worried that mRNA actually is going to get into your DNA. Doesn't happen. mRNA is actually transcribed in the cytoplasm of the cell by ribosomes that are in the cytoplasm, and it never makes it into the nucleus. There is no integrase. So you can't actually integrate the mRNA into DNA in the cell. Furthermore, we're only in macrophages, and those cells are going to die within 48 hours of having phagocytosed something. So these cells don't stick around long. What they do is once they've phagocytosed this vaccine is they traffic to the lymph node. What lymph node are they going to? The axillary lymph node, so right under your arm. And in fact, one of the things that's happening is people, women especially, are going into their physician and they're worried that they've got breast cancer because they're feeling swelling in that lymph node under their arm. It turns out that that swelling, of course, is all of the immune cells coming into the lymph node to respond to the vaccine. So you don't have to worry, it's not cancer in this situation. If you've been vaccinated, that's the swelling of your lymph node so that you're making cells to respond to the vaccine. Some of the spike protein is released extracellularly, and that will stimulate B cells to make antibodies. Some of that spike protein is presented in MHC molecules so that we can stimulate our T cells to produce interferon gamma and develop CD8 T cells, which will go back and kill any virally infected cells. So that's how our mRNA vaccines work. Now, the Pfizer mRNA vaccine, does it cause production of antibodies? Indeed, it does. And when they did these studies, they looked at several different doses of mRNA. The dose they settled on for the vaccine that made it to market is 30 micrograms, and that's this collection of peaks here. And what you can see is after the initial dose of 30 micrograms, we get a little bit of antibody. After we give the second dose with this little caret is, you can actually see we make a lot of antibody to this vaccine. But remember, antibody is only protective when the virus is extracellular. So do we make T cells? And the answer is we do make T cells. So here's our 30 micrograms again, and you can see 10 out of 10 people are making CD4 T cells to this vaccine. If we look at 30 micrograms for our CD8 T cells, 80% of people, 8 out of 10, are making CD8 T cells. And what we see over here is that those T cells are actually making interferon gamma to the receptor binding domain. So we actually get our Th1 response with interferon gamma. Let's look at the Moderna vaccine. Do we see the same response? Moderna shows their data a little bit differently, but let's look at the antibody response first. And the dose of antibody the dose of mRNA that's being used in the Moderna vaccine is 100 micrograms. So if we look first at the antibody titer, you can see that we get a great antibody response with that Moderna vaccine. And if we look at the antibody to the receptor binding domain, again, with 100 micrograms, great response. Now, you may have heard that Moderna said, well, you could just use half a dose of our vaccine so it would go farther. Because look at what they've got here. 25 micrograms gives almost the exact same response as 100 micrograms. So indeed, half a dose would probably work just as well. The reason that they're using a higher dose is that when we deal with elderly people who might be in immunosenescence, a larger dose is actually better. They also show that these antibodies can actually neutralize virus, and that's what they're doing here. All right, the Johnson & Johnson vaccine, which is coming to market next, this is an adenovirus vectored vaccine. So what they've taken is a human cold virus. Adenovirus is just something that causes a cold, and they've added the gene from coronavirus. So let's look at how this works. We start with an adenovirus, and we start with our SARS-CoV-2. We take the RNA for the spike protein and convert it to DNA. And then we put that DNA into the genome of the adenovirus. So now we've got just the spike protein, nothing else from coronavirus, in our cold virus. We also take our cold virus and we make it replication deficient, meaning that it can't replicate. It can't reproduce itself. So we've just got the only virus that you're going to be exposed to is what gets injected into you. It's not going to cause a cold. Then we do the same thing. We inject the virus into your deltoid. It gets taken up by macrophages. In this case, because we have whole virus, it actually gets taken up by a receptor on the surface of the macrophage. That's going to stimulate our danger signal. We see production of the spike protein from coronavirus, which stimulates coronavirus specific T cells. And the spike protein is also released, so we get antibody production. Simultaneous to that, we will be making an immune response to this adenovirus because our immune system sees this as foreign too. So you can see that we'll also be producing in our MHC molecules antigens that will stimulate adenovirus specific T cells and adenovirus specific B cells. So slightly different strategy. And I think I just showed you the data that the Moderna virus creates spike proteins. It does, because we're getting all sorts of antibodies to those spike proteins. Okay, so with Johnson & Johnson, again, we're going to present our data a little bit differently. What they did is they separated the data out by age so that you can see that here's our antibody, ELISA. We're looking at SARS-CoV-2 spike protein antibody. And again, with our various doses over various days, we get a very strong antibody response. We get a slightly less antibody response in those over 65. And so this is what brings down the stats for the Johnson & Johnson vaccine, is that we're getting a lesser response in the elderly population. But it's not no response, and it's enough of a response to neutralize. So this is a neutralization assay. It is enough of a response for the average elderly person to go ahead and neutralize virus. So good job with this virus as well. Do we get our CD4 and our CD8 T cells? And indeed we do. So we can see CD4 T cells both at day 15 for our low dose and our high dose, but not so much for our placebo. And same is true for our elders. We see at our low dose and our high dose, we see good CD4 T cell response. We see a good CD8 T cell response for those up to age 55. But if we look at our elders, we're getting a slightly less CD8 T cell response, both with low dose and high dose, which is again why we get 76% efficacy as opposed to 95% efficacy with this virus or this vaccine. The AstraZeneca vaccine is very similar to the Johnson & Johnson. The difference is that instead of using a human adenovirus as a vector, they are using chimpanzee adenovirus as a vector. And they actually compare the response to the Menactra vaccine, which is the vaccine for meningococcus, because that is a vaccine that we give adults. So what they're showing here is that here's our COVID-19 with just a single injection, and let's compare it to the Menactra vaccine, and we actually get a much larger immune response than we get to one of the vaccines that's on the market. If we give a boost, we don't see that much more immunity. So that's why single injection is what's being recommended for the adenovirus vaccines. The boosters are not doing that much more. Again, if we look at production of interferon gamma, we see a lot more interferon gamma production than we do for the meningococcal vaccine, and the booster doesn't give us that much more. The Novavax vaccine is a recombinant nanoparticle vaccine, meaning they're taking the recombinant proteins and they're actually hooking them to a nanoparticle to create kind of a larger antigenic complex. And they're using a new adjuvant that's called matrix M1. So we'll talk about this adjuvant as well. So let's look at this data. If we are getting antibody response, what we can see here is at 5 micrograms, here's our antibody response. And this is if we use our matrix M1 adjuvant. If we don't use the adjuvant, if we use matrix M1 adjuvant at dose 1, but we use a placebo at dose 2, you can see we don't get nearly the response. The adjuvant is necessary, is what the end result of that data suggests. And the same thing is true with the neutralization assays. Again, the adjuvant is necessary at 5 micrograms or 25 micrograms. With adjuvant, we get a decent viral neutralization. When we are using 25 micrograms, but no adjuvant, we don't get the same level of neutralization. So we need to have this adjuvant. What is the adjuvant? The adjuvant, historically in vaccines, our adjuvant has been alum, which is a form of aluminum. But as I think everyone is aware, nobody likes injecting aluminum into people. And so there have been adjuvants that have been tested for the last 25 years, and this is one of them. It's actually a saponin, which is a lipid, from the Chilean soap bark tree. So this is a natural product that's being used as an adjuvant to drive a Th1 response. And as you can see here, with no adjuvant, we get some response. With adjuvant, we get a massive response. So just adding some lipid from the Chilean soap bark tree, thank you, Chile, then we get a much greater response. All right. I was holding out a lot of hope for this vaccine, the Sanofi GSK vaccine. It's a subunit vaccine. And the reason that I was excited about it was that it doesn't generate a huge immune response. And so for people who might have autoimmune disease or things that are, more severe diagnoses, maybe tissue grafts and those sorts of things, I was thinking that, maybe a vaccine that isn't quite so strong would be better. However, this vaccine did not perform well in the initial trials. And so as of December 11th, Sanofi has gone back to the drawing board. It actually worked great in people who were 18 to 49 years old, but there was an insufficient response in people who are elderly. And so they are now going back and reevaluating what they can do to make this particular vaccine a little bit stronger. So here's where we're at. We get an antibody response with all of these vaccines, meaning that if the virus is extracellular, we can block it. We get a really strong T cell response from the Pfizer and Moderna vaccine, slightly lesser response from the Johnson & Johnson and AstraZeneca, and Novavax has not presented their T cell data, so I can't tell you what that says. Are these vaccines effective? They have been effective at neutralizing virus in ideal situations. So yes, all of these vaccines have actually been shown to be effective as of right now. So what's in them? Well, one of the big questions with the mRNA vaccines is what else is in there? And the adjuvant is actually the lipid, as I mentioned. But in both mRNA vaccines, polyethylene glycol is there. In the Pfizer vaccine, it's written out as, and in the Moderna, they abbreviate it, but it's the same thing. This is what we think people are having an allergic response to. So there have been cases of allergies to polyethylene glycol in the past. And so when people are having allergic reactions to this vaccine, it's not to the mRNA. It's probably to the lipids. Why do I say it's not to the mRNA? It's really hard to have an allergic response to a sugar. And that's what mRNA is. It's a sugar. And you can have an allergic response to a lipid. It's less likely. Usually what we have allergic responses to are proteins. So it's been kind of interesting to observe that folks are having allergic responses here. And the big advantage of these vaccines is because they're so easy to make, they're very easy to change. So as we have mutant strains appear, it takes roughly a day to turn around a new strand of mRNA. Now it takes closer to 2 weeks to grow that mRNA up, and probably closer to 6 weeks to get it out to market. But Moderna has already started working on mRNA vaccines that will address the new mutants that are on the market, that are in the population. The Johnson & Johnson vaccines and the Nova vaccines have not yet been approved by the FDA. And because they are not yet approved, they don't yet have to publish their adjuvant and their other ingredients. And so I can't fill this in. What I can tell you is that because this is a live viral vaccine, the adenovirus is a live viral vaccine, replication deficient, there will not be an adjuvant in it because any adjuvant kills the live virus. So there won't be any aluminum in there, and there won't be an adjuvant. There may be some other ingredients that will show up once we see FDA approval for it. So let's talk about safety now. We already know that some people are having allergic reactions to these vaccines, and the allergic reactions are happening at a fairly low rate. And anytime we do vaccine development, we require that the vaccines only generate a small level of allergic reactions. We're seeing roughly 2.5 people per 1 million. In other words, and I heard about another one a couple days ago. So we're at 11 allergic reactions, anaphylactic reactions in 4 million, I think we're at 5 million people that have been tested here. And again, the allergic response is likely to the PEG. That is not to say that there are not other side effects. Allergies is one of the side effects. People are developing headaches, fatigue, and muscle aches. This is the immune response, right? Headaches are being caused by the interferon alpha. The fatigue is being caused by interleukin 1. The muscle aches are being caused mostly by IL-6. So the fact that people are having these symptoms just means that their immune system is activated. The cytokines are what cause the symptoms. Remember that re-exposure should cause a faster immune response that is also more vigorous. So the fact that people are having headaches like within 2 hours of vaccination with their second dose just means that they've got a much stronger secondary immune response happening. What's happening for the folks who don't feel a darn thing? Well, those people typically are highly stressed and their immune system is suppressed. And so they're not feeling as many of the symptoms simply because the immunosuppression is keeping it low. Alternatively, they are on medications. And remember, there's pretty much no medication that doesn't suppress the immune response. So making sure that you're actually getting a strong immune response is going to be important. And you may want to get a titer if you didn't actually have any symptoms of vaccination. What about autoimmunity? This is a major question that people have. Are the mRNA vaccines safe for people who have autoimmunity? And do they cause autoimmunity? So first, let's review autoimmunity and how we develop autoimmune disease. You have to have T cells and B cells that have escaped tolerance. You have to be on a genetic background that you can present the autoantigen, and then you need an environmental exposure in order to get an autoimmune disease. And when we get an environmental exposure, like a virus, we can either have bystander effect, meaning we activate some of those autoreactive cells that escaped tolerance with interferon gamma because it's present. And we do see that interferon gamma is present when we vaccinate. The other mechanism is molecular mimicry. Molecular mimicry says that we have similar sequences in the virus that we have in our own human proteins. And so when we attack virus, we also attack human proteins. So are the mRNA vaccines safe for people with autoimmunity? Well, we do know that they give interferon gamma production. If people are stable on autoimmune pharmaceuticals, this probably isn't going to have an effect. We probably are not going to see a flare. But if people are not stable on their medication, then yes, we may see a flare in people with autoimmunity. So this is where someone with autoimmunity would want to consult with their physician to see, are they stable on their medication? As far as do the mRNA vaccines cause autoimmunity, let's answer the first question of, does COVID cause autoimmunity? And this is a paper that came out on January 19th. We don't know yet whether COVID causes autoimmunity. Keep in mind that from when the first autoreaction happens in a human, it can take anywhere from 3 to 18 years to develop an autoimmune disease. What this paper shows is that there is some molecular mimicry happening here. Take a look at the antigens from the COVID virus across the top, and they only looked at 4 antigens in this paper. And then these are the human antigens down the side. And you can look at the percent reactivity in these columns. So we're going to expect 100% reactivity to SARS-CoV-2 because these are SARS-CoV-2 proteins, and we see that. We get 100% reactivity across the top. Now, if we look at the percent reactivity to other human proteins, what we see is that there's quite a bit of reactivity to mitochondrial antigens. And you might be thinking, oh my God. But remember, this is antibody activity. Mitochondria is intracellular. So unless your mitochondria are extracellular, these antibodies are not going to reach the mitochondria unless there's cell damage. So in somebody who might have lupus, where there's a lot of cell damage, we might see some autoreactivity happening here. But otherwise, I'm not worried about the mitochondrial antigen per se. There are some other antigens, however, for which we see some significant cross-reactivity. So let's just look at the envelope protein as an example. We go to the percent cross-reactivity. We scroll down here, 56%. What is that? Beta-catenin. So here's an example of potential cross-reactivity with beta-catenin. What about nucleoprotein? Scroll down. Here's 45.5%. Insulin receptor. So we've already seen that there is more diabetes happening with COVID. Is this one of the causes? Is this some of the cross-reactivity that we're getting with diabetes with COVID? And now let's look at that spike protein, because that's what we're actually using for these mRNA vaccines. And so we've got the mitochondrial antigen. We already talked about that. And then there's this protein here. This is called neurofilament protein. So this is a protein that is in the CNS, the central nervous system, and we do see some cross-reactivity with it. Remember, though, that this is one person who has antibodies that cross-react between spike protein and neurofilament protein. We don't know yet what percentage of the population is making the antibodies that cross-react. Could this lead to some sort of autoimmune reaction? Indeed, it could. We would be naive to think that it couldn't. Are we seeing that autoimmune cross-reactivity yet? No, because as I said, it's going to take somewhere between 3 and 18 years before we know how much autoimmunity is actually going to be stimulated by coronavirus, much less by the vaccines. Fertility is another big concept that people have been worried about. And one of the big questions is, do the mRNA vaccines cause infertility by stimulating production of antibodies that bind to the placenta? So the concept is that there's a shared sequence of amino acids between the SARS-CoV-2 spike protein and a receptor that's typically found on the placenta. And first of all, is that true? It is true. There is a shared sequence. It's 4 amino acids long, and it is the membrane spanning region of the spike protein that is shared. The theory is that antibodies to the spike protein could then bind to the placenta and cause miscarriage. And there is no evidence to support this, but it is a good theory. So let's look at the evidence. Well, there were 44,000 women so far in the COVID epidemic who have had successful pregnancies, who have had COVID and then gone on to have successful pregnancies. So it didn't happen in them. Does that mean that it couldn't happen in anyone? No, it doesn't mean that. But the reason that we don't think that this is going to happen is that the shared sequence is so short that the likelihood of cross-reactivity is small. Usually, in order to get cross-reactivity, we need at least 8 amino acids, and we're only seeing 4 shared amino acids. And so again, the likelihood is very low, but the fear is out there. And so now you can dispel the fear because we know that it probably is not happening. What is more likely happening is the actual infection is actually been shown to cause infertility in men. So COVID-19 has shown to cause infertility in men who have had COVID-19 in roughly 30% of males. And that was a Lancet paper that came out a few months ago. Not in everyone, and it's not permanent, but it does happen for a little while. What about these variants that are hitting the market? So one of the variants, the mutation that's happening in the UK variant, is actually increasing the number of spikes per virus particle. So if you think about how you look at a virus particle and you see how those spikes are all spread out, turns out this variant actually increases the number of spikes so that they're more dense, which means that they are more infective. And it was probably naive to think that more infective viral particles were not going to cause more death, because since they are more infective, they get into more cells, and therefore it requires a much larger immune response in order to fight that. And it's often the immune response that is killing people. One of the other mutations that is happening in the spike protein, and it is at the furin cleavage site in the S1 protein. And it makes it so that the spike stays intact longer. So again, we've eventually our immune response just breaks down this spike protein. But when we take out that furin cleavage site, it hangs around longer. But here's what I want to tell you about mutations. There's been more than 1,600 variants that have been shown to exist just since the last, 7 months. Each lineage, each of these 1,600 variants is gaining around 2 mutations per month. We are going to have variants. There are going to be lots and lots of variants. So we can't let that derail us, because they're going to show up. And the reason is that we have a lot of people who are getting sick who are in immunosenescence or who are immunodeficient. And when they have immunosenescence or they're immunodeficient, then the virus hangs out in their body for a long time, and a lot of virions circulate. And because there's a weak or absent immune response, lots of mutations can happen. Then we give them antibody therapy or some other therapy, and that provides a selective pressure that eliminates anything that the antibodies can bind to and just allows those mutated viruses to survive. And that's how we wind up with mutants. There's also cellular reasons that these viruses mutate. There's enzymes that will do substitutions, and it creates less stable base pairs, which are not recognized by our pattern recognition receptors and make them less likely to stimulate an immune response. Just a couple of comments on COVID risk. I'm just going to talk about the top 3 here. We already all know that comorbidities, gender, and age have an effect on risk, but let's talk about the genes. And I bring this up because we keep hearing about, young, healthy people who have severe COVID, and they must have some underlying way that they're not healthy. Not necessarily. There are several genetic variants that have been shown to get more severe COVID. Specifically, there's a genetic variant on chromosome 3. There's a genetic variant of the interferon alpha receptor 2 gene, and that is raising the risk of severe COVID by 30%, and it's found in a quarter of people of European descent. So some of these variants are actually pretty common. And this makes people who are not elderly, are not immunosenescent, have more severe disease. And this is one of the things we worry about. The microbiome is also impacting whether or not people get severe disease. In particular, we see that people who come in and have severe disease are depleted in certain species of gut bacteria, like Faecalibacterium prausnitzii, Eubacterium rectali, and several of the Bifidobacterium species. Now, there's a problem with this. This is epidemiological study, and it's correlative, right? We don't know if this is cause or effect. We don't know if people already had deficiencies in these bacteria and that's why they're getting bad COVID, or if COVID actually caused the deficiency. So when we see correlative data like this, we pay attention to it, especially because we're seeing that this dysbiosis persists after clearance of COVID. So now we want to think about giving prebiotics that might stimulate the regrowth of some of these species. You'll notice I said prebiotics, not probiotics, because probiotics are not permanent. Prebiotics cause these cells to grow again. Vitamin D status is getting a lot of press because there have been studies out of France and Italy and Indonesia that suggest that vitamin D status is related to disease severity. By far, the strongest study was out of Indonesia. However, I need to point out that when JAMA went looking for the Indonesian study, it turns out that the authors couldn't be found and the hospital didn't exist. So this paper turns out to probably be false. It probably was just created. And that, unfortunately, has led the government to mistrust the vitamin D data. Currently, there are 73 studies on clinicaltrials.gov, but if these studies are not funded by the NIH, they're having trouble being trusted. And this is crazy because let's just take a look at some of the studies on clinicaltrials.gov. Told you there were 73, I'm just showing you 6, but look who's conducting them. Here's one that's being conducted by Harvard, right? Brigham and Women's Hospital in Boston. And the problem is it's internally funded. And so because it's internally funded, it's not considered trustworthy. I think we're going to have to get over our distrust of who's funding these studies. The big thing to remember is that if you're going to get a study funded by the NIH, it takes anywhere from 6 months to 2 years. So the reason these studies are not funded by the NIH is that these folks wanted to get these studies off the ground right away. And you're going to have to get some other source of funding at that point. All right, why is there so much emphasis on a vaccine? Why don't we just do herd immunity? I mean, I'm an immunologist. I should believe in herd immunity, right? And I do. But let's start with some of the misquoted data out there. First of all, I've heard that the death rate isn't any higher for the last year than it was for previous years, and that's not true. The death rate is higher for everyone except people under 25. And we think that the death rate is down for people under 25 because there are less accidents. And this tends to be the highest cause of death for people under the age of 25. Other than that, we have the highest death rate that we've had since 1949. So what are we talking about for death rate? Well, in 2019, it was 86,000 people per 1 million. Now we're at 97,000 people per 1 million. And you may think that's not that many, but that's per 1 million. So let's take the number of million of people who are over the age of 25, which is roughly 200 million adults, and take this number and multiply it by 200. And now we start to see that our death rate is actually much higher than it was in previous years. In previous years, we would say that roughly 8,000 people per day die. And now we're seeing closer to 11,000 people per day dying. If we look at basic epidemiology, that tells us a little bit more about why we might not want to just get herd immunity. In basic epidemiology, we take people who are susceptible, and some percentage of those people are going to become infected, and some percentage of those people will become immune, and some percentage will die. The big problem with COVID is that it's a little bit different than our other infections. So those who become immune, if we're talking chickenpox, you become immune and then you're immune. But if we're talking COVID, you become immune and then a large percentage of those people are going on to have additional symptoms. When I say a large percentage of people, what am I talking about? Well, we're starting to reach the numbers that we saw with SARS. With SARS, we saw that 40% of people went on to develop long-term consequences of disease. And we're seeing that now with COVID, that roughly 40% of people are going on to develop disease. So that's why we don't want to recommend herd immunity, because herd immunity from getting a COVID infection alone could lead to a huge public health crisis. We don't have enough physicians to treat all of these side effects. The other thing is that vaccines are a public health measure. And when we think about medicine, we're thinking about the individual. When we're thinking about public health, we're thinking about the population. And we make different decisions when we decide to live in community than when we live isolated. When we live in community, we have to pay attention to how our health affects everyone else's health. And that's when we have to weigh different risks. So COVID has its own sets of risks associated with it. The largest risk is death, but for a large percentage of the population, the side effects, the long-term side effects, are equally a big risk. For vaccines, it would be naive to say there's not a risk associated with having a vaccine. There will be side effects, there will be people who die, but we see less death than we see with COVID. We see less side effects than we see with COVID. And our goal is to protect others when we vaccinate. Our goal is actually not protecting ourselves. I know that that's what we have people doing right now, is rushing out to protect themselves with a vaccine. But really what you're doing with vaccination is you're paying attention to you and then all of the people you're exposed to. So I'm not going to tell you what your decision should be on whether or not you should vaccinate. You need to weigh the risk for yourself, and you figure out whether or not vaccination makes sense for you. That's what I have, and I'm going to open it up for Tabby to answer or ask me some questions. And I'm sorry I talked a little longer than I intended, Tabby. Sorry, Dr. Parker. Should I be calling?
Oh no, Tabby's great.
Okay.
Of course, that's great. So first of all, I just really want to thank you. This reminds me of my days as a student in your class, and just being, just so in awe of what you know. Honestly, Heather, it's great to just see this presented in this way, in this balanced way, and just really to go back and look at some even of the basic concepts that we forget about, so this has been really great. We have a lot of questions, so I'm just gonna start in the Q&A from Richard Hammerschlag. Hi, Heather. What can be done to mitigate the often reported flu-like 24-hour side effects of the second vaccine shot? And you did talk a little bit
Yeah.
about why that is, right? Why we're seeing more reaction in that second shot and how that makes sense. Just ideas around that.
Absolutely. So I will tell you it's different in every country. In the United States, they're recommending that you take something like Tylenol or Advil. The UK is recommending paracetamol. In Asia, they're recommending curcumin. And curcumin is actually working very well for reducing the inflammation that is causing the side effects that we're seeing from that second dose.
Great. Could you speak at all just to any of the research that you've seen or thoughts on vitamin C? I know that this is an area that our community is very interested in, especially.
So COVID depletes your vitamin C levels. And so there are 56 clinical trials right now on vitamin C, both as IV therapy, as oral therapy, in a variety of different ways in many, many different countries. Some of them are completed, but the data hasn't hit the market yet. But yes, vitamin C is something that is depleted when people have COVID, and it's such a good antioxidant. And we know that oxidative stress is one of the major issues that people with COVID have. So vitamin C is likely going to hit the recommendation status as soon as those trials get reported.
Great. Another question, let's see here, just questions about those who have had adverse reactions to corticosteroids and epinephrine, or anaphylaxis to other medications and foods. Do you have any suggestions if a certain vaccine has less or more possibility for that, or how to approach that? And this might be the, you know, as well, just any questions that are specific to your own personal condition. I mean, you really do have to work with your healthcare provider to think about what your risks are and, you know, where you fall in that range. So
absolutely. So absolutely having the conversation with your healthcare provider. What I would say is that if you have had anaphylactic reactions in the past, both Moderna and Pfizer have said that you should probably avoid their vaccine, and you might want to wait till the Johnson & Johnson vaccine or the Novavax vaccine, because at least in the phase 1 and phase 2 clinical trials, there were no anaphylactic reactions actually in phase 1, phase 2, or phase 3 for either of those vaccines. So I would wait and don't do Pfizer or Moderna. Wait until one of these other vaccines hit the market.
Yeah, there's another question similarly around someone being treated with immunotherapy for melanoma. 2 doctors have said it's fine to get COVID vaccine. A third has said to wait for J&J. Where's the best information and choices for those being treated for cancer? Do you feel it's safe to get the mRNA vaccine?
Yeah, so what's really interesting from the cancer perspective is where you are in the treatment process. If you're getting chemotherapy or radiation currently, you are immunosuppressed. So at that point, I personally would say I'd wait to get the vaccine until I was done with my therapy, right? Like we vaccinate a herd so that those who have cancer are protected. That's kind of the whole philosophy around herd immunity. Once you have gone through your chemotherapy and your radiation or surgery, the various treatments that you're going to have for cancer, then I would say any of those vaccines are actually going to be fine because they drive a Th1 response, and the Th1 response is an anti-cancer response. So it's actually going to help you fight any tumor cells that the chemotherapy didn't get.
Great. Hope to
Thanks
hear
for
your
doing that. I forgot to stop sharing my screen.
Oh, that's okay. Any knowledge, do you have any knowledge about the Brazilian strain of COVID and the vaccine outlook for combating strains that are not currently effectively vaccinated against?
I don't have a lot of knowledge about the Brazilian strain. They have started publishing those papers. The WHO is publishing them. I just haven't had time to read those.
And anything, do you know about the Sputnik Russian vaccine at all?
Yeah, it's an adenovirus or a vectored virus vaccine again. I'm not sure it's adenovirus. I have to double check, but their numbers are fantastic. They are with their vectored virus vaccine getting numbers in the range that we're seeing with the mRNA vaccine. So like 95% effectiveness. So it's quite effective. Now, again, anytime we see data coming out of other countries, we always have to double check it because some countries have government influence on the quality of data that is allowed to come out. And there could be data that is negative that has been suppressed. So we just have to pay attention to what's coming out, make sure it's consistent with what we see when we test it in the United States.
Thank you. Is there a post-vaccine follow-up or antibody test to make sure our body has optimal response? I have also heard some people are getting the antibody test after they get vaccinated to see what that is. So just any, what's your outlook on this? And
yeah, it's a great question. So, and I've had a number of friends who have gotten antibody testing after they've gotten their vaccines as well. And some of them have been positive and some of them have not. Doesn't mean the vaccine doesn't work. You've got to remember that the antibody tests for COVID were developed for antibodies to all of the different proteins, and you're only going to be making antibodies to spike protein for the vaccine. So it's entirely possible that you could show up antibody negative after you've been vaccinated, even though you've got plenty of antibodies to spike protein and plenty of T cells. But if you really want to test yourself, just go get one of the antibody COVID tests and maybe it'll show up positive for you and you'll have a reason to celebrate.
Another question from Dr. Hanaway, how is the body able to break down the modified mRNA? Do the RNase enzymes work the same for the synthetic vaccine, or will it stick around for a long time?
No, the mRNA is rapidly broken down. In fact, they've shown that when it's not in the lipid that's protecting it, it's broken down just as fast as mRNA that your body's making. And I've seen questions about this, but RNases kill it. It's why you actually have to freeze the Pfizer vaccine at -80, because if you don't, the RNases just kill it immediately. So the mRNA is still broken down. There was a second half of that question though, Tabby, I missed it.
Oh, and let me pull it back up here. The other one was, do the RNase enzymes work the same for the synthetic vaccine, or will it stick around for a long time?
No, they work exactly the same.
Great. What about women with breast cancer who've had lymph nodes removed for treatment? Are the mRNA vaccines still effective?
So if you've had lymph nodes removed, keep in mind that at any location, you're going to have multiple lymph nodes. So you should be fine. Even if you've had breast cancer surgery and you've had the axial lymph nodes removed, there should still be enough cells there to mount a response. But if there's not, your immune system is very wise and it goes to the next lymph node, which is the brachial. So you'll just move down your arm and you'll have a reaction in the brachial lymph node instead of the axial.
Great. What was the T cell response with Moderna vaccine?
They see a very good CD4 T cell response. They don't show us in a graph. It's actually in text in the paper. And in the paper, by the way, they do report that some people had allergic responses. Even though they didn't show us the data, there was a sentence that was hidden at the end of a paragraph that said some people had allergic responses in their clinical trials as well.
Are certain vaccines, so you did speak to this, are certain vaccines preferable to others for patients with autoimmune disease? And you did speak to this a little bit.
I did. And I think that, you know, the Novavax vaccine is probably where I would go with somebody who has an autoimmune disease, or Johnson & Johnson. I probably wouldn't go straight to the mRNA vaccines only because there now are other options. But if you are stable, the MS Society actually came out with a statement saying that if you're stable on medication, they recommend even the mRNA vaccines for people with autoimmunity. So, so less worry about the autoimmune disease if you're stable. If you're not stable, talk to your physician. Get stable.
How permanent is immunity?
Sorry. That's
Right,
such a hard question. It's such
exactly,
a hard
right?
question.
Right.
So, but let's talk about this in the way that we can. Okay, we don't know how long the immune response is going to last to COVID. We don't know how long the immune response is going to last to these vaccines. We're a year in. We're not even a year in for the vaccines. So here's what we can do. We can compare, just like they did with the Menactra vaccine that AstraZeneca did. We can compare the response we get to some other vaccines to what we're getting to these vaccines. If we do that, what we see is that the response that you get to the mRNA vaccines is more similar to, say, a tetanus shot than it is to, say, a flu vaccine. So whereas flu vaccine, we have to vaccinate every year, we're getting a stronger response to these vaccines, and it suggests that we might have longer term immunity. That said, if the virus mutates, that immunity may actually be irrelevant. We may have to get revaccinated anyway. So it's kind of one of those things like playing the lottery. I have a hard time going on record saying, you're going to have 10 years with this vaccine. I don't know that.
What are your thoughts about the latest plan to study mix match of vaccines? And speaking with a colleague who's an MD vaccinologist, she's concerned it could prime the immune system in one direction and different direction with the second vaccine from a different type, causing problems with immune response theoretically. So she just goes on to say, we don't know enough about priming. What are your thoughts about this?
If you're looking at exactly the same spike protein sequence like we do with the Moderna vaccine and the Pfizer vaccine, I'm less worried about mixing and matching. If you're using a different sequence that might be used, say, in the Johnson & Johnson vaccine or the Novavax versus the mRNA vaccines, then yes, mixing and matching wouldn't be recommended until we actually look at it.
Mm-hmm. That makes sense. And what are your thoughts about Johnson & Johnson requiring only one injection and that they'll have 100 million doses available in short order? Do you anticipate that they'll become more predominant given the availability, the easiness of logistics versus the mRNA?
It's hard to say. One of the availability issues is really the number of vials that are out there for people to put vaccines in. Another availability issue is the number of syringes that are out there. And Johnson & Johnson is going to hit the same issues with those things as any other company is. Mm-hmm. So maybe, but I don't know. But I'm totally fine with the Johnson & Johnson vaccine, especially for anybody 55 or under. I mean, we saw as much as an immune response with that vaccine for people in the 18 to 55 group. So I'm not too worried.
And that just ease of one shot will definitely,
that would help a lot.
That would help a lot, sure. Mm-hmm. Thank you, Heather. This is so useful. Can you talk a bit more about mRNA being a carbohydrate sugar? I thought that RNA was comprised of nucleotides and proteins.
So it is with a sugar backbone. So the backbone is made of sugar, and then yes, you're going to have, so I use sugar also interchangeably, like it's ribose nucleic acid, right? So that's the sugar anytime there's an ose. But yes, there are some proteins in there, but it's not very much protein relative to the sugar. It's a sugar that we're looking at there.
Do we know what adenovirus is being used by J&J? If we use a previously ubiquitous virus, would we then destroy it too quickly with memory immunity?
It is known which virus is being used by J&J, but I couldn't tell you that off the top of my head. But the strain, like the DNA sequence has been published. I mean, this is known. Could you destroy it too quickly? actually, I think they're counting on that, because ultimately what we want is we want you to mount a fast immune response to a vaccine, especially when we have a pandemic. And so my guess is that's actually one of the reasons that they chose this virus. A is that it's not dangerous, and B, because we already have immunity to it, our response to it will be fast. So it doesn't necessarily mean that the response to the spike protein will be as fast unless you've already been exposed to COVID. But my guess is that was part of their strategy.
Are there different levels of antibody or T cell response for folks who have already had COVID-19 or who are long haul survivors?
Probably. You know, and this is one of those things where we are in a very complex time, right? If anybody listened to NPR this morning, there was a whole story about the number of people who really have already been exposed to COVID versus the number we think have been exposed to COVID. And so when someone is driving through a drive-through vaccine at the San Antonio convention center, we don't give them a COVID test to see if they've already been exposed. So, I mean, this data, it isn't out there yet, but it will be. But likely, if you've already been exposed, you're going to mount a much larger response to the vaccine, which means you will have a bigger headache. You might have some more muscle aches, and it may last a little bit longer for the first injection, which is making a lot of people scared to get the second injection, because essentially their first injection was having COVID. And so the first injection of vaccine is actually their booster.
That's right.
So yeah, so that absolutely could be happening right now.
So there's a question: if someone had a small amount of hives immediately following the Moderna vaccine, like their first shot, would you agree with the contraindication for dosing the second?
That's right. If you have hives from the first shot, that's a Th2 response, which means a second shot could lead to an anaphylactic reaction.
Is it possible for the adjunctive adjuvant matrix M could potentially stimulate an MISC type picture?
I don't know.
Let's see here. If you have dysbiosis in SIBO, doesn't that give you an immune issue, and a vaccine may give you more symptoms, or is it worse to get COVID with poor immunity?
Yeah, I mean, there it is. That's the big question. That's the one that everybody's weighing, right? That if you're already sick, should you get a vaccine, or are you at higher risk if you get COVID? And the answer is that you're probably at higher risk if you get COVID than if you get a vaccine. And the difference is that, you know, we can predict that you're getting a vaccine, and so we can actually help mitigate that risk with physicians knowing exactly what's going on, versus you get COVID and you have dysbiosis and you die because we didn't know that that's what was happening. And by the time that you're in the hospital, you're on a ventilator and nobody can tell what was happening for your body. I mean, we just don't know. Like, this is where we want you to you have to weigh your own risk, and I can't make that decision for you.
That's right. That's right. So a couple more questions. I know we've gone over a little bit, and I just really appreciate you taking this time with us. Are there any studies on the immune system or microbiome on people who are COVID positive but are completely asymptomatic? Like that difference? Not yet, right?
Not yet. Yeah. Because the only way that we do these studies is when people come into the hospital. So if you're asymptomatic, you're not coming into the hospital. And so no, not yet.
Can you comment on the use of relative risk versus absolute risk when it comes to determining the efficacy of mRNA vaccines?
No.
Okay, thank you.
I mean, yes, I can, but it's mathematical, and I would actually have to draw it out, and I'm sorry, can't draw it out right now.
Not for a webinar that is right.
Not at this moment.
Any recommendations for COPD patients who are on chronic inhaled steroids?
Yeah, so again, if you're on chronic inhaled steroids, likely you're not going to have a massive reaction to a vaccine because the steroids shut down your immune response. So the folks who are on steroid medications are probably going to have a very safe vaccine response. They're also not going to mount a massive immune response such that that uh, we don't know how much immunity you're actually going to get from the vaccine, right? Because the steroids are going to shut that down. So it's safe to get the vaccine. The question is whether or not the vaccine will be effective.
And I think this will be our last question before we close out. I'm scheduled for my vaccine on February 11th. Can I fly the next day? There's been a death in the family.
Yeah.
This is something I have to do. Is that contraindicated at all?
Not contraindicated. The vaccine does not cause spread. It is not infectious. You'd still need to wear your mask while flying, right? Because you can be vaccinated and still spread COVID. What vaccines do is they decrease the symptoms and they decrease the amount of time that you're infectious, but it doesn't mean that you can't still get an infection.
Yeah,
So,
can you just repeat that one more time?
Yeah.
Right, because I think there are some misconceptions too, just around what this is going to then mean.
Yeah.
And like how this is just one step in the bigger, approach, really a public health approach that is wide and varied, not just focused on one thing.
Absolutely. So what a vaccine does is it primes your immune system such that if you get exposed, your immune system is ready to respond at an instant. That means that if you get exposed, the duration that you have COVID will likely be shortened. Instead of 3 weeks that people are having COVID, you might have it for 5 days or 3 days, or you may be completely asymptomatic. Doesn't mean you can't still spread it in that first window when you're exposed. So we're still going to have to wear some masks while COVID is circulating, even if you've been vaccinated, because it doesn't mean that spread stops. It means that it's reduced over time. And because it's reduced over time, as more and more people are vaccinated, then spread stops. But just because you got vaccinated, it doesn't mean that you are somehow safe and you can now go maskless and go, out spreading your lovely germs to everyone else. It means that you've reduced the window where you might be infectious.
And hopefully the severity a little bit.
And definitely the severity. Yeah, that's what we're seeing with all these vaccines is it brings down severity like 0 hospitalizations. That's fantastic, right?
Fantastic. So those folks who are at risk and really, that's where they become more protected as well.
Absolutely. And, I can't tell you how many people I've talked to because people keep contacting me about questions that they've had, but like I've talked to a family that had to go to the hospital because their father, the patriarch of the family, died, and everybody came out of the hospital with COVID. We need to get our hospitals back.
Yep.
We need to have people be able to visit their sick relative who, had a heart attack and not come out, and now the entire family is infected. So that's really kind of essential.
I have one other question. I've had a lot of folks ask me about the difference between this and say a flu vaccine, and like each year there's a different flu vaccine. Will that potentially happen with COVID? What will, could you speak to maybe what that could look like?
Yeah, so we know that COVID, that the SARS-CoV-2 virus is not as mutagenic as influenza. It is more mutagenic than measles. So it's not going to be one of those things where it's like measles, you get a vaccine once every 18 years.
And that's it.
But it doesn't appear to be as mutagenic as influenza yet, where you need a vaccine every year. So it's somewhere in the middle. And I don't know at this point where it is because we're just so early in this pandemic.
And that's one of the things, right, that we'll have to watch and look and study and see then what direction we have to go in.
And keep in mind, just like we've had really bad flu years, in fact, 2019 was a bad flu year. We had a strain of flu that circulated that was a strain B, and people got really sick with flu. And then we've had flu years that haven't been severe at all, right? It could be that that's what happens with our coronavirus seasons, is that there will be years that are severe and there will be years that are not severe at all. We just don't know yet. What we predicted prior to this 2020 was we have seen coronavirus epidemics happen every 10 years. We had SARS, we had MERS, and now we have COVID. So that was what was predicted, is that every 10 years we would have a coronavirus outbreak. No one suspected that this year's coronavirus was going to go pandemic. So again, we just don't know.
That's right. Well, any closing thoughts or comments or message to our community that you'd like to leave us on, Heather? I just so appreciate you.
I think the only thing that I would say, because this is an integrative health community, is don't forget all of those things we already know. Sleep. Sleep is so important for resetting your immune system. Make sure you're getting enough sleep. Nutrition, so important for immunity. Make sure you're getting your nutrition and keeping your gut healthy, you know, and all those other things we know, meditation, herbal medicine, massage, like take care of yourself, because we know that that's going to really help us all, not only for surviving COVID, but for having the mental health to deal with all of the other BS that comes with it.
Great.
Yeah.
Well, it is always such a pleasure to learn from you. And I just really thank you, Heather, again, for giving this time to our community and to the larger community. I just will end out by thanking you all for joining us today. I'm sorry we weren't able to get to everyone's questions, but we're just really happy that everyone showed up, and hopefully continue coming back to our wellness webinars each week. What's next? Next, this is Heart Health Month. And next week, we'll be having a Heart Health in Focus with Dr. Mimi Guarneri, who is our founding president and a integrative cardiologist. And we're very excited about that talk. So join us next Friday at noon for that. And I just want to remind people that if you're thinking about joining our AIHM fellowship, we have an actual deadline today for a mental health professional scholarship. So please take a look at that. You can visit aihm.org/fellowship or email fellowship@aihm.org to ask if you can still apply for that scholarship. Our next cohort starts in April 2021, and we still have a few spots available. So if you're interested in really getting high-level training in integrative health and medicine, please contact us. And we are offering a $500 discount to the fellowship if you attended this webinar and apply in the next week. And stay connected with the Academy. So many of you have asked, can I get this video? I really want to, I wasn't able to attend today. All of our wellness webinar videos are free to the public. We post them on our YouTube channel. So if you sign up to youtube.com/AIHM Global, you'll be informed when we upload the videos. You can also find us on all the other social media platforms. Please consider joining the AIHM member community. We are an interprofessional community of integrative health and medicine providers, and we would love to have you as part of our community. So thank you again, everyone, and we look forward to seeing you next week. And again, Heather, thank you so much for this great presentation.